Hey, so good afternoon, everyone, and welcome to this parallel session on discovery neuroscience. And thank you for joining us. My name is Tomás Ryan. I'm a professor in neuroscience at Trinity College Dublin. I'm also the chair of the Advocacy Committee for FENS, the Federation of European Neuroscience Societies. We represent about 30,000 practicing research neuroscientists and their students across Europe and affiliated countries of the European Union. Our interest here is to advocate for the importance of fundamental discovery neuroscience, what it takes to do it, what kind of barriers we're facing, and crucially, the impact that it is having and can have on society. What we heard this morning in both sessions was the importance of brain health, not just for general productivity, but for innovation. The brain is the motor of innovation. And for society, but also for health, understanding how the brain works and understanding the basis of brain disease is necessary for us to make progress. And we also heard very good examples of initiatives that have been provided by the European Commission and also specific countries in order to improve our understanding of the brain and in order to engage with issues of brain health. What we have not heard much of yet is the impact of research on our understanding of the brain. How fundamental research has, in the past few years, created new insights, which has allowed us to interpret what is happening in the brain, to change how we understand the brain, in order to make new advances in how we orientate research programs and how we treat disease. And I think it's very important, this point was made earlier about public communication, that in order to make neuroscience, in order to make brain health relevant to citizens and to policymakers, we have to be able to explain in plain English what is it that research has done to change our understanding of the brain and to thereby have a positive impact, have a positive contribution on society. I think to make this point, we have an absolutely ideal panel of speakers. I will briefly introduce the four of them. First, we have Urté Neniskyte from the University of Vilnius in Lithuania. We have Balázs Hangya from the Institute of Experimental Medicine here in Budapest. We have Tomás McWilliams from the University of Helsinki in Finland. And then lastly, we have Attila Kaszás from TTK, also here in Budapest in Hungary. They're all early to mid-career researchers working in discovery neuroscience, and some of them on the border of discovery neuroscience, preclinical and applied work. Fundamentally, they represent much of what the next generation is of discovery neuroscience research. And they've all made proven contributions to fundamental neuroscience over the past 10 to 15 years. So I'm going to ask each of them three different questions before we open the discussion to broader topics. First, we're going to ask about their research. Then we're going to discuss what success may look like for their specific research fields in the next 10 to 15 years, assuming that they have the right support, that society is invested in these questions. And then thirdly, we're going to talk about some of the specific barriers that we may be facing as discovery neuroscientists. So I'd like you all to engage with the first question in order. And the question is, tell us briefly about your research and about some contribution from the past, from your recent career, that you think has made an impact on how we understand either brain function or disease. Starting with you, Urté.
Thank you. Thanks a lot for the introduction, Tomás. So I'm a biochemist from training. And I got into neuroscience during my PhD studies. So the way I look into the brain, I usually think about the molecules and the interactions there. And I started looking into the brain from this perspective during my PhD studies in Cambridge with a particular question: how do the immune cells in the brain interact with neurons and change the networks. In that case, it was in the pathological context. Now it kind of moved on to look in physiological context as well. And what I was very happy to contribute to was the very first study, which actually showed that when our immune system gets overactive in the brain, it can recognize neurons that are not dead, they're just unhappy, but recognize them as targets, remove them, thus contributing to the neuropathology and contributing to the elimination of cells that could otherwise survive. It was very difficult to convince the society, which happens with discovering neuroscience, a community in a society, a community that it's actually happening. But since we have demonstrated that and published that indeed immune cells can eat alive neurons, these effects have been seen in different contexts, such as development as well. And why I think it's important, because when we did hear just now, just before the coffee break, about the traumatic brain injury. And traumatic brain injury or stroke are one of these cases where limiting this eating of neurons that are just unhappy and not really dead yet can limit the pathology, can limit the extent of the injury, and it's actually allowing better recovery. So my PhD supervisor, whose ideas these were, is still continuing on working and actually moved into the drug discovery to actually look what are the molecules that could actually interact with this. But the looking for drugs only became possible now when you already know that this process is happening and we already know the molecular pathways that actually are required for immune cell and neuron interaction.
Okay, thank you very much, Urté. And indeed, intersections of the brain and the immune system is a continuously important topic in current neuroscience. So, Balázs.
Thank you very much. So we work on the neural mechanisms of cognition, so the neural mechanisms of everything related to thinking. And most importantly, we work on the mechanisms of learning. We also work on how this goes bad in different types of dementias. But I want to stress that already by working on how learning is happening in the brain is the same as working on dementia, because we won't understand these diseases unless we know how it works. And this must be very obvious, but somehow this message got diluted throughout the years. But I mean, if you take your car to the repair shop, you may say, I don't care about batteries and catalytic converters, just go and fix my car. But someone inside there must care about the battery and the catalytic converter, otherwise the car won't be fixed. So going to what we discovered about learning, well, I will argue that to understand learning, we have to understand how the brain reacts to things that we find rewarding, and how the brain reacts to things which we find aversive, and actually things that predict those things that are rewarding or aversive. So we must know how our brain reacts to these cues. And we did discover as first how the famous cholinergic system in the brain reacts to rewards and punishments and things that predict these. And this kind of started a field which looks closely into how different cell types are behaving in this situation. And I would argue that this will be fundamental to understand how learning works. But if you want something more tangible, I can also say we actually discovered some cell types which are differentially affected in Alzheimer's disease, and now we understand their genetics, so now they can be made specific targets for therapy.
Thank you very much, Balázs. And as a memory researcher myself, I think the insights your group has produced have been quite crucial for understanding processes of learning, and indeed in the current world, we're starting to be able to parse how the processes of learning and memory is, and to understand them as very separate biological substrates. So Tom McWilliams next.
Well, thanks very much for the opportunity to participate here. So my lab is firmly at the intersection of what I would call discovery and translational neurobiology. And what I mean by that is that we study what was once regarded as a very abstract and fringe process. So in plain English, my lab studies how the body breaks down components, right? It's a process called autophagy, to eat oneself. This was regarded as a sort of luxury belief in the 1980s because it was studied largely in yeast cells with electron microscopy. It then, through sustained investment and effort, flourished. And in 2016, the Nobel Prize was awarded to Yoshinori Sumi in physiology or medicine for its predicted importance. And so it's a fascinating topic because our brain cells have to live for decades and they need quality control mechanisms to survive. And so our contributions to the field have effectively been lighting up this process. So previously people could only study it in cultured cells. And my mentor and I made tools to actually visualize landscapes of destruction inside intact brain circuits and indeed breaking out of the brain into other tissues. And possibly something that I think vindicates the primacy and importance of discovery research is that in 2022, I think it was, we published with our collaborators in the New England Journal of Medicine the first ATG7 deficient patients who were thought to actually could never exist. And this demonstrated the clinical significance of a phenomenon that was, previously viewed as fringe. And therefore, I think it's important to say that nobody has a monopoly on serendipity. Nobody knows where the next discoveries come from. And I think the research field that I'm involved in is emblematic of that. So yeah, we study how things are broken down. We try and light it up and look at their significance. And we work with everything from patient cells and clinical collaborations right through to collaborations on bats and yeast and mice. So we just go where it's interesting to discover, but we're constantly anchored back to the physiological and pathomechanistic significance of the process. That's what we're captivated by.
Thank you, Tom. I think a wonderful example of how tool building and new methods have been crucial for opening doors to, for unconventional ways of investigating mechanisms. And then Attila.
Yeah, so I'm Attila Kaszás, and I actually started here in Budapest before moving to Marseille, and now I'm back to Budapest. My work involves modern novel technologies. Primarily at the beginning I was working with imaging technologies, but regarding epilepsy research. But during the time spent in France, I found that they might be a bit inadequate to give the proper answers. So I moved into the field of neurotechnology, and one of the major results that we actually managed to achieve is a probe that allows the modification of populational activity in the evoked epileptic neuronal populations, stopping epileptic activity actually. And continuing on that line, when I moved back to Budapest, right now we are looking at glioblastoma research, which is in connection with epilepsy in multiple ways, because glioblastoma is actually the primary marker for glioblastoma is the sign of epilepsy in patients. And we plan to use neurotechnology for detection and modulation of events in the cortex. So it actually is more a pragmatic approach towards basic neuroscience, and how I connect to the discovery neuroscience is basically finding out how we can adapt our neurotechnology methods to fit the brain modulation and the observation of brain function. So now we are involved in a study that would actually define what are the biohistocompatibility parameters to use a device which would be actually capable of glioblastoma modulation. And for that we plan to use both microfabrication methods and the previously developed imaging and neurotechnology methods.
Thank you very much, Attila. Very inspiring stuff. So what we've just heard from the panel are a set of concrete examples for, I think, what we can regard as the credibility for basic discovery neuroscience. It's not simply necessary. If we don't have discovery neuroscience, we won't have any clinical or translational neuroscience. But we're starting to see, particularly, I think, in the past 10 to 15 years, largely because of new methods becoming available, the strides that we are able to take in our understanding of the brain. And so this leads to me, now that we have some, I think, base credibility established, my next question for each of the panel is, what does success look like for these subfields of neuroscience? And if you want to go further for neuroscience in general, assuming that we had no realistic barriers on our ability to get on with the work, so to speak, starting with you, Urté.
I'm in the wrong place, aren't I? I think in molecular biology, not just in neurobiology, for all the 20th century, we were over-focused on proteins. There's one gene, one protein. You mess with one gene, you mess with one protein. It's quite straightforward. But again, I'm a biochemist, so that's not just proteins in the neurons. There are also sugars, there are also lipids, and the research of my lab and also of many others shows that those sugars and those lipids are very important when we talk about the establishment of the networks, refinement of the networks, both at the neuron-to-neuron interaction and neuron-to-glia interaction that I'm in particular interested. And one of the examples is our recent paper where we didn't really look at neurons, but we look at glioblastoma cells, and we showed that modifying the sugars on the glioblastoma cells changes how they form the network within the brain tissue. And this research was actually possible because we had the support of the patients who did have neurosurgeries, and they allowed us the secondary use of the brain tissue. And then we used the glioblastoma cell lines established again from the patients to see what happens if we modulate those sugars. And we saw that we might have situations, we might model the situations where we either get very dense and very well integrated network of glioblastoma, which we know from the research of others is a problem, because that means that it will have very strong epileptic activity and the glioblastoma will be much more aggressive. But we can actually block this process and not allow glioblastoma to form these networks, making them much more accessible for other treatments with other drugs. So this is just one of the examples, again, coming from a very fundamental point of view, trying to understand how surface sugars modulate the network, how they how actually this can show you the new druggable target. So now getting to your question is that with new technologies, again coming from the discovery science, such as gene editing technologies with CRISPR and other tools, I think we are now in the place when they actually modulate very different pathways. And we can start understanding the brain comprehensively from different points of view, accounting for different macromolecules and understanding how these macromolecules interact in developing brain, in physiology. If we talk about memory, we need refinement of networks, in the pathological conditions. And supplementing this with multi-omics, we have spatial omics, we have metabolomics, actually having this biochemical background that defines how our brain functions or misfunctions, and then understanding how we can target it to alleviate the diseases.
Balázs.
So extrapolating from where our field is already heading, I imagine that it's gonna be actually a lot more holistic, while still being, of course, fundamentally reductionist, in the sense that, well, now we focus on how small dissected pieces of the brain work, almost not in isolation, but not considering many factors we cannot at the moment control simultaneously. But I think we're gonna go in a direction where one needs to understand the genetics, the physiology, the brain-body interaction, and neuroimmunology kind of at the same time and in the same context. This will actually require a lot of a very diverse expertise for the projects, which I imagine will stretch the limits of the small groups doing hypothesis-driven science. So it may actually also require a somewhat new format, but that I think will work itself out. I think this is largely driven by the needs, which is good, and that's where I think we are heading at the moment.
Tom.
Yeah, I think for our field, it's about theranostic interventions. And what I mean by that fancy buzzword is therapeutic and diagnostic intervention. So we would love to have a minimally invasive method to monitor when, where, and how turnover pathways or degradation become stalled in humans. And we're getting there. Most of the healthy aging studies that have been conducted with respect to neurobiology, they've been on short-lived model organisms. So things like yeast, worms, flies, which are incredibly important and tractable. However, there are different selection pressures compared to longer-lived mammals. And so about 3 weeks ago, we just published our longitudinal brain aging study on autophagy. And what was really fascinating was that we found that midlife was a critical period for how these pathways are regulated in the brain, and in contrast to the short-lived model organisms where they go down. And it depends on the cell type and the region, but in mammals they can actually increase and be sustained throughout life. And so we were really gratified when our chief of neuropathology in Helsinki reached out to me and said, oh, by the way, we've conducted one of the largest autopsy studies ever completed on 700 brains for healthy aging, and we actually find midlife is where a lot of the early neuropathology seems to manifest. So actually, I'm super excited because we're getting a handle on at least the when bit of the equation. Now, how to monitor that in humans and in cohorts at scale and sensitively, that's going to require a good bit of brain power, but I'm confident we can get there. And ultimately, it would be to defeat degeneration. I think that's what really is our guiding light. But yeah.
Yeah, so I guess regarding our research, where neurotechnology is involved, we are sort of in an outer position because we are relying on everything that has been discovered previously to actually fit the brain functions and to make the technology compatible with the previous discoveries. In the future, how I imagine what we could move forward to is to have a device that would be able to stay in a patient for a longer period of time and maybe help in the treatment of different diseases such as the reductionist approach to glioblastoma, so actually making glioblastoma a bit making the treatment of glioblastoma possible. That would be a further aim. For that, we would need to rely on the on molecular tools to actually have the compatibility properly set, and also to have the physics properly set for the given protocols for the neurotechnology device that we are using to have the treatment possibility in the future. And how I believe that actually on the wider global field of view, how the neurotechnology can help brain health is there's multiple examples nowadays how brain-computer interfaces can interact with the brain itself, and how it can they can improve brain health and improve the life of patients. There are really a lot of examples that are in the media nowadays regarding applications of neurotechnology for paraplegics, making them walk again, for giving vision or at least part of vision back to the blind. So that is a further aim, but definitely not within 10 to 15 years of range, but we hope to get there soon.
Thank you all. That is just such a positive and yet grounded vision of where things can lie in the near future. And I don't think it takes a lot of imagination to see how that will impact brain health. I mean, I'm a neuroscientist, and I often tell my students that I'm a neuroscientist, and that's just another way of saying that I don't understand how the brain works. And it's a feature of how we communicate the brain, not just to society, but to each other, is that even though I'm a neurobiologist, I actually have greater and more comprehensive insight into how a kidney works, how a liver works, how the digestive system works, even how the immune system works, because I know that science knows these answers. And as a general biologist, I can give satisfactory explanations to how those organs work to a science student or to a layperson. And we struggle with the brain. We struggle in explaining how we go from this organ in our skull to complex behaviors. And I think the pictures that you're all painting give us a canvas to work with for imagining how things can be different in 10 to 20 years as new generations of researchers start to make progress and provide insights that bring big data together, that bring larger projects together such that we can make sense of it. But my last question before we go on to more broader questions from the audience is, what do you feel as an individual, as a young research professor working in 2024, are the main barriers towards making this progress? I mean, we know that science is slow, science is hard, but we also have many external barriers. Some of them are manifest in our universities, some of them are manifest in our countries or funding systems. Some of them might be the absence of things that we wish were there to enable us to engage with research in a more creative way, in a more efficient way, or in a more collaborative and global way. So I'd like to invite your comments on this issue, and we can do it in the same order. So, Urté.
So I'm probably thinking about two major barriers. One is the lack of funding for bottom-up research, because as Tomás already said, nobody knows where the next discovery will come from. And when most of our funding comes from a programmed research, we're only investigating things that we already know. We're just going deeper, which is important, but we're not giving enough space for new discoveries. And since we don't understand how brain works, and we don't really know where the secret lies, we do need to have enough funding for bottom-up research. If we want to think outside the box, that's what I've heard today, patients saying, we need to think outside the box. The only way to think outside the box is to have sufficient funding for that. Another problem is actually quantitative assessment of researchers. Everybody recognizes this is a problem, but last year I attended an exercise at our university which was trying to move from quantitative to qualitative research, trying to give numbers to qualitative research. So we pretend that we're doing qualitative, but we still want to measure that somehow. So that's, again, it's not specific to neuroscience. I think that's valid to any research, but we need to rethink how we assess scientists, in particular because science is slow and science is hard, and sometimes it takes very long to actually get to the answer. And if we say to the person, you just need to publish now, they will publish, but they will not go as deep as they could, as their brain allows them actually to look at the problem. So these are the barriers that I would like to see overcome over the next 10 years.
So I actually just would want to go one step further than Urté and say, in general, I think the biggest problem is inefficient mechanisms. So just as often people tell scientists, well, you have to look at the big picture. Also for how we run society, we have to look at the big picture. Just let me tell a few examples. When there's a small problem which someone thinks it can be fixed by a new checklist, when you introduce a new checklist, well, also think about what you want your trained scientists to do with their time as a society. Or if you launch a new grant with all the goodwill, if the success rate will be 3%, and if you calculate just the time the community will invest in writing the grants, it might be actually a net loss for society. And bad metrics for evaluating science is part of it, because if you ask scientists to, and if you make it very competitive and say, well, I'm gonna measure you like this, then everything else you don't measure, well, why would one do it? You can say, well, that's your moral obligation. That's usually the answer to this, but that can become a shaky ground. So, and you may argue efficient mechanisms is expensive to set up. Of course, it takes thought, but I will argue inefficient mechanisms might even be more expensive. Just throwing more money at it and hoping that it will resolve itself instead of putting all the thought in it.
I agree with all of the aforementioned points, and one thing I would like to add is I think we're at risk of developing an amnesia for the heritage of innovation and how this happens. So a lot of the things that we're really excited about in precision medicine and translational medicine, they were never intended for this purpose. So if you think about iPS-derived cells, modeling using patient-derived cells, this was never ever intended for this. I mean, it was developmental biology research, which was the genesis of this. The same thing for, Urté mentioned CRISPR-Cas9, genome editing. There's so many examples. So I actually think a latent and sinister threat is to assume that we know where everything is coming from and that we can direct serendipitous, impactful discovery. And that is important for the political understanding of science. So we often as well talk about communication and the public understanding of science, and that's incredibly important. But I also think the COVID-19 pandemic sort of emphasizes the importance of policymakers and bringing them into the fold as to why we're doing what we're doing. And curiosity-driven research could lead to cures in some instances, but it's okay if it doesn't, because we could actually have new technologies that are disruptive by nature, and nobody could have predicted that. I think if they did, they had a different crystal ball than everybody else. Yeah.
Yeah, so I would like to, first of all, acknowledge all the points. Primarily with the inefficiencies, I think that I could agree with that. I think one of the main barriers is the lack of permanence in research. Many times, just building a lab requires at least one permanent researcher, plus all the staff and all the researchers that would be helping a project. If a project is run for a given time, usually that knowledge is lost when that particular researcher moves to another lab. This is actually a grave danger regarding neurotechnology, because in these projects, many of the knowledge is done by multiple people. Microfabrication by engineers, measurements by biologists. If the team is disassembled during a project, this will lead to inefficiencies, meaning that though devices will be produced, but the end result will be not beneficial for the society because it's just an intermediate product. So for that, for the funding of these intermediate and continuous projects would be quite beneficial, because then they would be able to underline the previously developed technologies and make fundamental research support whatever usage they might be, whatever advantage they might be having for the society. So I think in that regard, a bit more permanence would do quite a lot of good to many of the projects.
Thank you all for the lucid description of the challenges that we're facing. And it's remarkable to see also that although I know these particular talking points were not pre-discussed, there's a complete agreement between 4 very different types of scientists working in very different places on the kinds of challenges that researchers must face on a daily basis. And it seems to me that if we're going to actually get around them, if we're going to improve this situation, it's going to require more and more collaboration with people working outside of science. And the point has already been made that the way science was mobilized so quickly in the COVID-19 pandemic is a clear illustration of how sensible collaboration with policymakers is absolutely crucial in order to, I suppose, move research to the next stage to move fundamental research in neuroscience to the next stage in its history, which I think is why forums like this are becoming important. We have nearly 10 minutes left in this session, so at this point in time, I would like to throw the floor open to the audience members to ask any questions they have of the panel, either relating to these topics or on other topics. So.
Yes, if I may start, Roger from Utrecht from the Netherlands. So I think I agree with everything you say, but if we want to make an argument that we need more investments, we need to convince politicians that more efforts need to be taken for discovery neuroscience. We have to come with something better, more tangible, and we have to identify those gaps in knowledge that will really result in new breakthroughs and game changes for addiction. So I agree, but I think you have to do better and come up with some generic measures in fields that need investment. Just simply say we need more discovery in neuroscience is probably not a convincing argument. What's your response there?
Who wants to take that?
That kind of comes back to the programming research. You know, with this approach, we wouldn't have CRISPR gene editing. With this approach, we wouldn't have RNA vaccines. That's the thing, you know.
You have to have a kind of rationale that you have to, first of all, a rebel mechanism is the first step to, get better treatments. And of course, we have to identify what we don't understand. We now know that there's at least 5,000 cell types in the brain. We got no idea what they do. We have a lot of genetic data. Most of the genes we got no idea what they do. So this is already a bit more concrete. And I think there's a few genetic things like also gene therapy is coming up. There's a lot of these technologies that need investment, and we have to specify what the major areas are in discovery neuroscience and in the technologies that need investments.
I found that when interacting with policymakers, and I recently had a conversation with one politician who asked me this exact question: how do we quantify that scientists are going to deliver on the kind of investments that we are asking for? And I was dealing with a politician from a relatively small country. And a lot of it was about illustrating how we actually work. And I had to say, as scientists, we generally go up for an election every 5 years. It's when we're renewing our major research grants. And the pressure to do that is immense. We have a built-in competitiveness that I don't think people outside of science are always aware of. And once I started speaking in that language, they were immediately understanding how a baseline productivity is calibrated, but it's very difficult to put a number on it. And I think what was illustrated by the discussions that were given by these 4 speakers is that it was a qualitative thing. I mean, the main impacts that have been made, including CRISPR, not only did they come from discovery neuroscience, but they're not something you can put a number on, right? There's something that emerged from very fundamental insights that people developed and that then have been carried by other people into other domains. Putting a quantification on that within a particular country or on a 5-year time horizon is going to, in my opinion, going to take us in the wrong direction for selling the work, because we'll end up with perverse incentives that are driven towards things that are not conducive to research. And I think it may be on us to persuade policymakers that we already have very clear evidence that what we're doing is working?
Yeah, I echo all of the points that have been made, and I think your question highlights a sort of different, more lateral problem, and it's that, you know, how do we dissociate discoveries which, as I mentioned repeatedly, cannot be predicted, right? Disruptive discoveries, truly things that are paradigm shifting. You know, how do we dissociate the importance of that and how those happen from an election cycle? And that comes down to our own advocacy as a community, right? And there are very, very good positive controls for this. We know, for example, that there is an important appreciation of discovery neuroscience, at least historically. I'm not speaking about the current climate in the United States, and the positive control for that is the fact that there is about 10x the investment over there, and this is a federal thing. It comes down to our perception of what discovery neuroscience can be used for. So if one looks at actually where a lot of funding comes from in the United States, it's the US Department of Defense, right? This is viewed as integral to security policy and to having a sort of strong and competitive economy. So I actually think your question is opening a tinderbox of complex things, and we actually need, as neurobiologists, neuroscientists, along the continuum of discovery to translation, we need a far more coherent and sophisticated advocacy strategy than we've been having. Because, you know, the general public, they accept the Large Hadron Collider, right? I mean, this is fundamental research at its essence, and it's a wonderful example of what can be achieved. We need a sort of similar strategy to do this for discovery neuroscience, and I think it will happen.
Thank you, Tom. Question over there.
Hi, David Mannon from Cambridge. Just looking for sources of funding. One beneficiary of all of the work that the 5 people on the panel there do are the pharmaceutical industry. Their net profit margins, I was just looking up on Google, is about 13.6% after taxes and everything else. This is what they take home. The global pharmaceutical industry, also looked up, is $1.6 trillion a year. If we ask them to increase the amount of tax they get by 0.1% and say that's going to be hypothecated for fundamental biological research, I think you'd find a much more generous acceptance of that than just saying we're going to put up the tax. In fact, you could go to the 4 or 6 biggest pharmaceutical industries, sort of partners, and say, we're going to put together a fund which will provide this pre-competitive research for people like those sitting on this panel. And I would have thought that they would see that as worth investing in. The amounts of money for each individual firm are small, which begs the question, is there nobody from industry at this meeting, or is there?
I would like to just respond, and I think you raise a fantastic point, and it's something that we often neglect as well. This is where the medicine is made, right? The pharmaceutical industry, for all of people's arguments for and against it, that is where the rubber meets the road for translation. Without them, we cannot do rigorous, you know, patient-impacting work. I think what you said is fantastic, and we should maybe discuss it afterwards, because, and you're right, industry should be part of this conversation. There's also phenomenal research done in industry.
Agreed.
Right?
I think this is clearly a case where we can look at the United States, as well as having very developed neuroscience funding. There is, I think, a clearer awareness of the circularity of these relationships between the pharmaceutical sector, between how much funding is given to the NIH, because it's understood that the pharmaceutical sector cannot work without taxpayer funding from the USA. In Europe, we simply just don't yet have that awareness, I feel.
Yeah, part of it is because Europe is many countries put together when you look at the size of the market. But just 2 other suggestions to make. From what Attila was saying, the other thing to think about as basic neuroscientists is to say, I wish I had a technology for this question. And then there's a small amount of funding for workshop produced by this process to say unanswered questions and unmet technology needs. And then that's put out, and you see what comes. It's very early stage research for technology development. And similarly, if you have a clinical context in which you might be able to answer a basic science question, So the point about looking at long-term network connectivity, again, the point you were making, implantable sensors after someone's had glioma surgery or epilepsy surgery, not to look at the primary disease, but look at network formation. And people have done this simple things like intracranial pressure measurement and so on. But with the advanced technologies, finding populations of patients in whom these relatively low-risk implantable sensors can be left would be a great way forward. So I think finding the nexus of needs and opportunities may be a really useful thing. Start off with workshops which are low cost, and then the opportunities can be evaluated and looked at.
Yeah, I completely agree that we need a bit more heavier connections to the clinics, and actually here in Budapest we plan to do this to connect to the clinical approach. And actually regarding the first part of your remark, we definitely would need to define the questions that we actually have devices that are giving the answers to. But many these different research groups, sometimes they are producing devices just for proving that the devices can be actually produced, but they lack the networking and they lack the connections to actually put it to good use. So going into that direction would definitely make these research projects way more valuable. And actually, what I was saying is that we would need to push into that direction for the intermediate designs and intermediate devices, so that we actually see the use for that. So I completely agree.
We've run out of time, so hopefully these conversations can continue later in the broader event. I would just like to thank our speakers from taking the time to travel away from their research groups in order to come here and make these contributions. We greatly appreciate it because we have to spend so much time in science evaluating each other. I think it's becoming increasingly important that together we also advocate for neuroscience more collaboratively with society, so that some of these barriers can be removed and we can increase the health of discovery neuroscience in Europe. So please join me in thanking our speakers.